Hi, welcome to Technical Tea Breaks. I'm Nadia Halidi, head of the Advanced Light Microscopy Unit at the Center for Genomic Regulation. In this video, I'll be presenting live cell imaging. So, why live cell imaging? Well, live cell imaging or time-lapse imaging provides an accurate study or accurate record to study dynamics, cellular processes, behavior and function in real time over time. It gives a more realistic view of biological function. However, it is much more challenging to image live cells, keeping them alive on the microscope stage and healthy over an extended amount of time. In this video, I would like to guide you through the most important considerations when designing a live cell experiment. These are, for example, sample preparation, choosing a microscope, whether it's inverted or upright, maintaining live cells on the microscope stage, efficiency of detection, photobleaching and phototoxicity, the ultimate limiting factor, which is your photon budget. So let's start with sample preparation. You've chosen your sample, you would like to mount it on something to put it on the microscope stage. Now for that, you have a number of options that are commercially available, but also you can do your own. For instance, you have multi-well dishes, small petri dishes as small as 35 millimeter with a glass bottom cover slide, or a multi-well slide, also glass bottom. But you can also take cover slide and mount it yourself with a 3D printer or 3D printer molds or something that you have bought commercially. You can opt between choosing either a transparent or a dark-walled one. You can also choose a flow chamber. This would enable you to hook it to a perfusion system to allow fluid passage, apply drugs or inhibitors while you are acquiring your image. You can opt between an open chamber or a closed one. And here in this example, you can see that there are multiple channels here, and you can add in drugs and inhibitors throughout your channels. This is called a microfluidics setup. Where you can use teeny tiny amounts of drugs or inhibitors or any media to study cellular behavior. But you can also create with microfluidics a concentration gradient to study your behavior as well. Sometimes cells don't like to adhere to whatever substrate you have. You want to enable this adhesion. For that, you would probably want to use extracellular matrix proteins, ECMs. Coat these substrates with the ECM, like fibronectin, collagen, and laminin, whichever, but you have to choose the one that is most appropriate to your cell. Once you've seeded your cells, you want to put imaging media on top. Avoid media with autofluorescence properties, those that contain phenol red, sedum proteins, because their background is huge and it will affect your signal to noise. Take into account whether you want to use a CO2 dependent media, which means that it would require CO2 in the atmosphere, or a CO2 independent media that requires buffers such as HEPAs, and that would maintain the pH in your imaging media. You would like to stain your cells as well. Follow dynamics of certain proteins or ions. For that, you would want to stain your sample. Use fluorescent protein tags, fluorescently tagged ligands, fluorescent antibodies to extracellular epitopes, cell permeant small molecule fluorophores, for example, dyes, DNA stains. The key thing is to look at two aspects: the brightness and the photostability of your fluorophore. These characteristics are written usually in the data sheet of the provider or in the manufacturer. Now, you've prepared your sample, you want to take it to the microscope. Which microscope would you choose? An upright or an inverted microscope? Now, usually samples mounted on a multi-well plate is very difficult to approach from the top. So you'd want to use an inverted microscope for that. Samples won't usually grow on glass bottom dishes. For that, you would want to use an upright microscope, maybe even a dipping objective. Your sample is too thick. Now that one is critical because it might be that neither an upright nor an inverted or configuration is good for you. You would want to choose something completely different, like light sheet, when you have the illumination and the detection is only from the sides rather from the top or the bottom. Then you might need to access your sample during acquisition by adding drugs or inhibitors, even though I would rather recommend you to use a perfusion flow or microfluidics setup. Then location of what you want to detect, what proteins perhaps you want to detect, adhesion sites, which usually adhesion sites are in the bottom of the dish or at the surface of the glass. You would want perhaps to use total internal reflection microscopy. For that, you have no other option. It's inverted microscope. Right, so you've chosen the upright or inverted or something else microscope, and you want to maintain the environmental control around your cell. So for that, the environmental control will control the temperature, CO2, and humidity. You have two options really. The first one is the whole enclosure, where within the whole enclosure, the temperature, CO2, and humidity is controlled, or a stage top incubator, which is very local. It's just that teeny tiny area. Each has its pros and cons, but the idea is the same for both of them, is to maintain temperature, CO2, humidity for your cells. You would have thought that now you are ready to image. Think twice. The efficiency of detection depends mainly on, first of all, the objective. Now, the objective determines the spatial resolution and controls the amount of light gathered from the specimen. Avoid refractive index mismatches between the sample and the immersion oil. This is very easy with live cell imaging. When you put media on top and you have your cell inside, you have a number of immersion, different immersion objectives. For instance, apart from the oil, which unless you're using it, unless your sample is right at the cover glass level, you would want to opt for something else or change the refractive index of the oil, immersion oil that you're using. You have the options of also water, silicone, and glycerin. You want to also consider the working distance, field of view, number of optical corrections in the lens design. I did not go through the details of the objective because this had been covered in another tea talk or tea break. Also, the refractive index is also have been covered in another tea break. So please have a look. Next then is the filter set. You want to know your fluorescent proteins absorption and emission spectra, and you want to also know the systems filter sets that it comes with. Filter sets had been also discussed in another tea break. Please have a look at that too. Then comes the detectors. Now, often for wide field systems and spinning disc confocal systems, you use cameras. Cameras can be CCD, EMCCD, SCMOS cameras. Depending on the brightness of the signal in your sample, you would want to compare those camera specifications in terms of sensor format, pixel size, field of view, read noise, full frame rate, quantum efficiency, and dynamic range. Line scanning confocal systems, however, they use photomultiplier tubes, typically multi-alkali photomultiplier tubes, but also they have the more sensitive ones, the ones that has gallium gallium arsenide phosphide GASP detectors. But also there are hybrid detectors, and a variety of them depending on whether you want to concentrate on resolution or sensitivity. One last thing that we need to consider is the illumination source. You would want to put this in the beginning, but here we are, it's here at the end. So illumination source could be for fluorescence imaging, could be mercury lamp. I wouldn't like to stress it, but mercury lamp. Also metal halide lamp, which contains some mercury. LED or lasers, different kind of lasers. And that all depends on what kind of imaging you would like to perform. The final image always boils down to signal to noise. Right, so we are ready to image. Two more things that are very, very common in live cell imaging. That's photobleaching and phototoxicity. Photobleaching is when you've exhausted your photo budget. The eyes are not happy. Cells are there, but there is no signal. You can't get signal out because it had died. You bleached your signal. On the other hand, you have phototoxicity, when the amount of light had actually made your cells unhappy. They started to behave differently. They started to form blobs, retract from the underlying substrate and detach, and ultimately can cause also the apoptosis. Now, generally, reactive oxygen species, or ROS, are fundamentally important for physiology as functional signaling entities. However, when ROS production is increased, it can cause damage of lipids, DNA, RNA, and proteins, and it induces apoptosis. So you need to maintain a very healthy level. Don't excite your cells with light too much. Right. We're talking about the photo budget here. You have to maintain, keep it, it's sacred. The iron triangle shows very nicely how to maintain this photo budget and how you can manipulate things. But three key things: signal to noise, temporal resolution, and spatial resolution. You have to find a certain balance between them, because if you want to increase temporal resolution, you will do that on the expense of the other two. Equally, spatial resolution, if you want to increase the spatial resolution, we do that at the expense of the other two as well. So you need to find a balance and make sure you decide on what you want to concentrate during live cell imaging. During the whole time, you want to maintain cell viability. You want to make sure that your cells are happy and alive even past your imaging session, that's past your acquisition time. Keeping this in mind, if you are keeping the triangle in mind, if you are working with live cell imaging, you want to expand this triangle a little bit more to incorporate sample viability and depth penetration. If you're taking into account these 5 aspects, which microscope should you use? Each of the microscopes has pros and cons. They are good for certain applications, not for others. Your unit staff members are the best people to talk to because they know in their core facility or core unit what kind of microscopes they have and what they are good for. Talk to them and discuss your project, and they will help you and guide you to which microscope to use. However, just if we touch base on how things look like, for instance, for wide field deconvolution systems or wide field system, temporal resolution is unbeatable, it's pretty fast. And if you go to the confocal systems, you have first, which performs better in terms of speed, is the spinning disk, and the least is the scanning confocal microscopes. Then you have multiphoton, which enables deeper penetration, and light sheet, which not just deeper penetration, but better temporal resolution. Again, talk to your facility staff members to help you guide you through these different microscopes and help you find the optimal microscope for your application. Once you're done imaging, acquired all your data, probably you've already thought of data management and storage. You want to process, analyze, and interpret your results. This is key for live cell imaging because otherwise you wouldn't be able to answer key biological questions. With that, I would like to leave you with some resources, and I hope you enjoyed the technical tea breaks.